Today we're getting into talking about viruses. We're going to talk about the first half of viruses today. Today we're mostly going to talk about the general properties of viruses and all things that all viruses have, as well as some important terminology for you. And then we're going to get into bacteriophages. And we're going to start our talk on bacteriophages. Monday, I anticipate that we will finish up our talk on bacteriophages and talk about eukaryotic viruses or animal viruses. So, first question is, what is a virus? How does it differ from most of the stuff that we've been talking about thus far? And most of the stuff that we've been talking about thus far, this being a microbiology class, have been microbes. What are some things that all of the microbes that we've talked about have in common?
Metabolism.
They all have a metabolism, right? They're all cells of one sort or another. Very basic things. They're all alive. Viruses are sort of similar to normal microbes and sort of different. They are arguably not alive. They're also not exactly non-living. They're sort of in between. They have a lot of characteristics that we think things that have these should be alive. Like they reproduce, they evolve, they pass on genetic information to offspring. These are things that non-living stuff tends not to do. This desk is not going to have any offspring. That chair doesn't pass on its traits, but viruses do. On the other hand, there are a lot of things that they don't do that we feel living things should do. They're not cells. They lack that very important cellular environment that is sort of the basis of life. While they can reproduce, they can't reproduce by themselves. They require help. Now, speaking personally, so do I, but I at least require help from somebody of my species. When a virus goes and looks for help, it doesn't look for Mrs. Virus to help it out. It looks for a host. And they don't have a metabolism by themselves. They do have a metabolism sometimes, but not all the time. So the general properties of viruses, they're not technically alive. Technically, they're not called organisms. They're called agents. You'll hear me talk about them like they're alive, and you'll probably hear me refer to them as organisms, and most people who study viruses do. But sort of technically, they're not alive. We talked about the 3 domains of life, right? What are they? Bacteria, eukarya, archaea, right? Which one of those are viruses in? None of them. So they're technically not within the 3 domains of life. All viruses are obligate intracellular parasites. What that means, obligate. An obligation is something that you have to do. So this is something that they have to do. Intracellular means inside of cells. And parasites means that they're going to live off of stealing something from a living organism. So all viruses have to live inside other cells and steal their stuff. Not all the time, but all viruses have to do that at least some of the time. Viruses don't possess a complete metabolism on their own. We'll get into this in a little bit more detail in a second. Viruses do possess a genome, they do reproduce, and they do evolve. All viruses have an intracellular and an extracellular state. All viruses spend some of their time inside of cells and some of their time outside of cells. Now, some viruses spend most of their time outside of cells. Some viruses spend most of their time inside of cells. But All of them do both. And viruses, as far as we know, infect pretty much all types of life. Now, we haven't actually gone out there and looked for viruses for every single species that we know. But every time we bothered to look, we found viruses. There are viruses for animals, viruses for plants, viruses for worms, viruses for flies, viruses for fungi, viruses for bacteria. I don't know if we've really looked for viruses for archaea. Yeah, I'm not sure about that, but there's probably some archaea viruses out there as well. So what are viruses made out of? Well, there's two things that all viruses have, and that is nucleic acid and protein. And these are two things that you have as well. What does your nucleic acid do? What is your genome made out of? We said that viruses have a genome.
Nucleic acid.
It's made out of what type of nucleic acid?
Diribose nucleic acid.
In the short form, that would be DNA, right. Your genome is made out of DNA, and your short-term instructions are made out of RNA. Viruses have a genome made out of nucleic acids. Their nucleic acids, their genome is a little bit more flexible. Your genome is tough. It's long-term. It's got to survive, because you're going to live a long time. Even if you're a bacterial cell, you're still living a lot longer than a virus. So your genome is made out of DNA. Viruses don't last for very long. Their genome can be made out of DNA or RNA. It could be single-stranded or double-stranded. So it could be double-stranded RNA, or it could be single-stranded DNA. It can be linear, like a eukaryotic chromosome, or it can be circular, like a bacterial chromosome. Most of them only have one chromosome, but not all of them. So there's a big, that's one way in which they're both similar and different. All formally living organisms have DNA genomes. Viruses don't necessarily have DNA genomes. It's always a nucleic acid, but it isn't always DNA. The second thing that all viruses have is a protein coat. And protein can do a bunch of different things. Primarily what protein can do is it can be an enzyme, and it can do chemistry. That's what we call a functional protein, because it does a function. Or it can be structural, meaning that it's going to give shape and structure and rigidity and toughness to something. What all viruses have is a structural set of proteins called a capsid. That's this thing on the outside here. I like to think of viruses as being sort of like M&Ms. They have sort of the gooey, chocolatey, soft nucleic acid center with the hard, crunchy protein outer shell protecting it, so that they melt in your body and not on your hands. Capsid. This capsid is this set of outer proteins, and it's usually made out of a bunch of proteins all working together. Those proteins that make up the capsid are called capsomeres. So a capsid is made out of individual capsomeres. That's what all viruses have. Then there's some stuff that some viruses have, but not all viruses have. Okay? Some viruses have what's called a lipid envelope, much like your cell membrane. It's a lipid bilayer with proteins embedded in it. In fact, it looks very similar to the cell membrane because most of them are derived from the cell membrane. Often they're specialized pieces of cell membranes that viruses steal on their way out. So this virus that you see here has a capsid, that's this sort of purpley thing in the middle, and it has the envelope surrounding it, and the envelope is made of lipids, just like your membranes. Often lipids with these specialized proteins embedded in it, and you can see that there's matrix proteins and glycoproteins labeled here. A virus with an envelope is called an enveloped virus. Makes pretty much sense. A virus without an envelope is called a naked virus. Viruses can be either enveloped or naked. The other thing that some viruses have, but not all, is enzymes. We already said that they have proteins. They all have capsid and capsomeres, so they all have structural proteins. Some viruses have functional proteins, enzymes. Not all, but some do. Often, this is going to be proteins that do things that the cell that they infect can't do by itself. So, for instance, a well-known member of that club is HIV. And HIV has to turn its RNA genome into DNA. What do you usually do? Do you usually turn RNA into DNA?
The other way.
It's the other way. You usually turn DNA into RNA. That's what you're good at. Since it has to go backwards, it has to carry its own enzyme called a retrotranscriptase, because it's kind of going backwards from RNA to DNA. It has to carry that in with it. So some viruses have some specialized enzymes that they take with them, and some of them don't. So that's generally what composes a virus. There are a lot of differences between viruses and cells. And I want to go over a few of those differences. This is a very good table from your textbook. So viruses are inert. Outside of a cell, they lack a metabolism. They only have a metabolism once they get inside a cell. Cells have a metabolism on their own. Viruses reproduce, but they do not grow or divide. Cells get bigger, and then they split somehow, and then they get bigger, and then they split somehow. There's a couple of different ways for them to split. Some of them we went over last time. Viruses don't get bigger. Once they're formed into their shape, they stay that shape. And when they reproduce, they don't split. They make kind of a bunch of different disparate pieces, and they have to be kind of put and hooked together and then turned on. So they're sort of assembled rather than growing in a kind of more organic process. Viruses aren't cellular. Cells are. If viruses were cellular, we'd probably call them cells. Viruses are obligate intracellular parasites. Cells are usually not, but there are some cells that are, especially some of the really small bacteria that are obligate intracellular parasites. We talked about the difference in their genome. It can be DNA or RNA. Generally, not both. They either have DNA genome or an RNA genome. We always have a DNA genome, and we always have RNA transcript. So their genome can be a variety of different things, double or single-stranded DNA or RNA, linear or circular. Our genome is always double-stranded DNA. Viruses are much smaller than most cells. Viruses range from about 10 nanometers, which is way too small to see even with the most powerful light microscope. It's smaller than light, to be entirely honest, to 400, which is something that, if it wasn't for the fact that the ones that are 400 nanometers long were only like 10 nanometers thin, you might be able to see. Cells sort of bump up against the high range here. They're going to go from about 200 nanometers, it's the very smallest cell, all the way up to 12 centimeters in diameter, which is, yay, that's like one of the bigger cells out there.
What cell is that?
I know.
So that is a weird type of protozoa called a plasmodial slime mold, which is actually a whole bunch of cells that all get together and sort of like Voltron assemble into one giant cell that's only got one cell membrane but has thousands of nuclei inside of it. And then They look like these big orange slimy things. You see them in forests sometimes. So viruses have a protein shell. Generally speaking, cells do not have a protein shell. And viruses, as I said, these are replicated in assembly line sort of fashion, whereas cells reproduce by division. So those are some important differences between viruses and cells. So viruses also have many different faces, and they go through a lot of different phases in their life cycle. Okay? And so all viruses have a part of their life when they're outside the cell and a part of their life when they're inside the cell, and they do things very differently. They're, in fact, almost sort of essentially different types of creatures in those two states. And some viruses have a third state, which we'll get into in a second. But I want to go through some names that we have for these different states. So a virus is always a virus, but depending upon the state that it's in, it might also be something else. Now, we talked about the protein coat, the capsid. When you need to, and admittedly in Arizona we don't wear a lot of coats, but pretend that this was someplace with a normal weather, like Boston, okay? Then you would put on your coat when?
Outside.
When you go outside. And when you walk inside, you're going to keep your coat on? No. Viruses are much the same. Coats are for protection. When they're outside the cell, they put their coat on. When they're inside the cell, they take their coat off, because they don't need it inside the cell. So a virion, or a virus particle, is a fully assembled virus with its capsid on. So like it's got its genome, and it's got its coat on, it's got everything it needs to go leave the cell. And it's, in fact, probably outside the cell. Generally speaking, these are metabolically inactive. So that's a virion, or a virus particle. A virus that's inside the cell is sort of just a virus, or a viral infection, and you can't really count them. We'll get into this a little bit later, but like if you see, like, a virus particle is a discrete entity. Right? And you could say, here's one virus particle, here's a second virus particle, here's a third virus particle, here's a fourth virus particle. When they're inside the cell, there's no boundaries between them and the cell. So you can't say, oh, there's 5 viruses in this cell. It's just the cell is infected with a virus. The virus is kind of everywhere. A last stage, and not every virus does this, but some do, is a provirus or prophage, depending upon whether or not it's working in bacteria. If it's in bacteria, we call it a prophage. If it's in eukaryotic cells, we call it a... Okay. So the provirus or prophage is when the virus has taken its genome and inserted its genome into its host's genome, so that there's no difference between them. Well, I mean, there is some difference, but so that they're a part of their host organism. They're integrated into its genome. They're now inseparable from it, to some extent. Often, but not always, they're dormant in this state, waiting for reactivation. Anyone have any questions thus far? All right. So let's start talking about bacteriophages. Phage is a word that means mouth or to eat. So bacteriophages are bacteria eaters. They were actually discovered before we really knew what a virus was. People started working with bacteria a long time before they could see a virus, because you can see a bacteria with a normal microscope like you guys use in lab. You're never going to see a virus with one of those microscopes. To see a virus, you need an electron microscope. But people could see the effects of viruses, And when they were studying bacteria, what they noticed was sometimes you would get this effect where no, she's just wandering by. Okay. Sometimes you would get this effect where something would eat the bacteria, and you would see these spots sort of start on the plate where the bacteria would kind of melt. That's awkward. Just sort of sitting there. And the bacteria would just kind of melt. And they knew that something was eating the bacteria, and they couldn't see what it was. So they called those phages. What it turns out that they were was viruses that eat bacteria. So this is your standard bacteriophage. And most bacteriophages look sort of like this. 2 out of the 3, and the pretty standard ones look like this. Kind of like some sort of, I don't know, depending upon how menacingly you draw them, they either look sort of like a moonlander or like some sort of demonic hell spider. Like, I'm pretty sure I killed these things in Diablo 3. They look kind of creepy. And they have sort of 3 parts. They have this head group at the top. That's what contains the genome. They have this bit here. That's the tail. They have a base plate at the bottom. And then they have these sort of tail fibers, which is these kind of leg-looking things coming out of it. And there's 3 varieties of bacterial phages: virulent phages, temperate phages, and filamentous phages. Virulent and temperate phages are similar, and they both look like that. Filamentous phages are completely different. We'll talk about them another time. All bacteriophages have a capsid and a genome. That's it. They're protein and DNA. They never have envelopes. They never have enzymes, at least as far as I'm aware. There's been a few that have been engineered to have them, but in their natural state, they don't. So bacteriophages are nice and easy because they only have a few different types of life cycles. And the first one that I want to get into today is called the lytic cycle. So all normal phages, all non-filamentous phages can do the lytic cycle. So virulent phages and temperate phages can both do it. Virulent phages can only do the lytic cycle. They can't do anything else. The lytic cycle, what does lysis mean? To cut. If we're talking about bacterial cells, when bacterial cells lyse, that means they're going to sort of blow up and explode all over the place. The lytic cycle is fast and aggressive. It's a good way to make a lot of bacteriophages, a lot of viruses really quickly, and it's usually pretty destructive to the bacterial cell. I mean, all viral infections are pretty destructive to the bacterial cell. But this one is destructive in a very quick and immediate way. So we start off, step 1 is attachment or adsorption. Okay? During attachment and absorption, the bacteriophage is sort of floating around randomly. Because bacteria or viruses outside of a cell don't have a metabolism. So they can't move on their own. They can't direct their movement. They have no flagella. All they do is randomly bounce around. Okay? So this virus is randomly sort of bouncing around, bumping into cells until its tail fibers stick to one of these cells. So it's sort of bouncing around back and forth, and boom, lands on that thing and sticks. This is called attachment. And the ability of a bacteriophage to attach to a cell is determined by the interaction between these tail fibers or legs and proteins on the surface of bacteria. Now, not every bacteriophage can infect every bacteria. They're usually very species specific. Like E. coli has the viruses that affect E. coli, and maybe a few very closely related species. Staph epidermis has the bacteriophages that affect staph epidermis, and a few very closely related species there. This property of the range of bacteria or the range of species that a virus can infect is called its host range. And the host range is determined in part by whether or not it can stick to the proteins on that cell's surface. Every different species has sort of unique proteins on its surface, and if the virus can't stick to it, it can't get inside. So this is one of the main things that affect host range. Going to step 2. Step 2 is penetration or entry. Bacterial cells have a cell wall. So, because bacterial cells have a cell wall, the entire bacteria can't get inside. The entire phage can't get inside. It's too big. So the whole virion can't enter in one piece. Instead, what it has is at the base of the capsid in that base plate, there are enzymes that chew through peptidoglycan. So what it's going to do is it's going to chew through the cell wall, and then the whole thing just sort of squats and compresses, just like a syringe. And when it compresses, it's going to insert this, this bit here, the tail, into the cell, and it's going to squirt the DNA in. It's going to sort of squirt its genome in, and it leaves the capsid on the outside of the cell, just sort of hanging around. It's now a dead shell. It could fall off, it could stay on, it doesn't matter.
In a previous slide, you said that none of them have envelopes or enzymes.
Bacteriophages?
Isn't that what we're talking about?
Yes. Did I say envelope?
Well, right now you just said that it had an enzyme that chews through the cell wall. Earlier you said they don't have any enzymes.
Good point. In this case, I guess what I meant was, I had to think about that for a second because I'd never actually put those two statements together. It doesn't have any enzymes sort of floating around inside here that get inside of the cell. But these enzymes are actually sort of built into the shell and don't go into the cell, but yeah. Okay. I'll have to think about that one and do some research into it and see whether or not those enzymes are a part of the structure or how they work. But that's a good point. Thank you for raising it. But they never have envelopes, to the best of my knowledge. Although probably somebody's going to point out something there that I got wrong. So, sorry about that one. So synthesis and replication is steps 3 and 4. Steps 3 and 4 are not very well divided. They both sort of happen at the same time. And what happens with synthesis and replication? Well, the first thing that happens is the bacteriophage DNA takes over the cell. And it says to the cell, hey, you shouldn't be making those bacterial proteins. You should be making viral proteins, because viral proteins are awesome. And so the cell says, okay, and it starts making some viral proteins. So you can see here it's just making capsid head groups and some tails, and it's making some legs. And pretty much at the same time this is going on, some of the genes that are being expressed from the virus are going to digest the genome. And we talked about this a little bit on Monday when we talked about generalized transduction, because generalized transduction involves the replication of phages. So the virus sort of degrades the bacterial genome, cuts it into a bunch of different pieces. And there's going to be a bunch of little pieces sort of left, but I took them away because unless you're talking about transduction, they don't get involved in this process. Right after that, the bacterial genome begins to replicate. And part of the reason behind digesting the host cell genome is so that the host cell can't make its own proteins. So the entire cell is now going to be devoted to making viral proteins. The other reason is it needs to liberate those nutrients that are locked in the bacteria's DNA in order to replicate its own DNA. So it's ripping apart the bacterial DNA, and then it's using those resources to manufacture its own new viral DNA bits. Did you have a question?
So does that mean like it's using those nucleic acids to build back up its own genome, or is that?
Yeah. It's taking apart the bacterial DNA, digesting it into more nucleic acids, so that there's a bunch of nucleic acids around for it to use. Step 5 is assembly or maturation. These words in parentheses are just, a lot of these stages have more than one name depending upon who's talking about them or what textbook you use. And the first word is the one that I usually use for it, but you might see the other ones as well. So I think of this as assembly. Some textbooks call it maturation. Either way, they both mean basically the same thing. And during this step, what you're doing is you're putting the viruses together, right? So you're going to sort of start assembling them piece by piece, and then you stuff the genomes inside of them, and you've basically got a cell with a bunch of, what are these things called? Bacteriophages, but more specifically, are they prophages? Are they just viral infections? Virions, right. Awesome, virions, or virus particles at this point because they're fully assembled. And you can tell me how many of them there are. How many virions are there in there? 4. If I go back here, how many virions are there in there? How many virus do you see? You can't really tell because it's all in a bunch of pieces. It's like asking me how many soda is in this bottle. Well, it's just sort of the soda fills the bottle. And in this state, sort of the virus is kind of filled